thank you to everyone for joining us, for being here. It's spring, we can finally be outside. It's a little less virtual than it used to be. So we're really grateful to the community for continuing to participate in our lecture series. I guess with that, it looks like we're leveling off a little bit for participants. So I'm going to go ahead and start to try and make us as timely as possible. You are at the 2020-2021 Virtual Community Lecture Series for the Sue and Bill Gross Stem Cell Research Center. I am Eileen Anderson, the director of the Stem Cell Research Center. Again, I'm really glad and grateful to everyone who's joining. I'm double checking to make sure there's no text coming through that tells me we have a problem in transmission. It looks like we're okay. And I just want to say a couple of remarks starting out. The first is a huge shout out and thank you, as we always have to do, first to Dr. Brian Cummings, who's our community outreach chair, also to Judy Beck, who runs our communications from the Stem Cell Research Center, and two particular folks at UCI Media who have just been so fantastic with us from the very beginning when we used to be able to do these in person and we're recording them and getting up on our website, Will Alvarez and Kyle Good from UCI Media. As you all know, we are a very diverse center, 63 plus faculty members spanning 21 departments and 5 different schools at UCI. Our mission is very much aligned with the School of Medicine here: discover, teach, and heal. So groundbreaking, basic science, educating the next generation of graduate students, postdocs, undergraduates, medical students, and so on, and also to heal. We have a very translational focus in the center, and that's a big component of what we do and how we think in terms of our science. You're going to hear that tonight. I promised when we restarted this series that I would keep my remarks at the beginning quite short, as opposed to all of the things we used to talk about back in the day as we were starting off this series in terms of educating people about CIRM and stem cells and California Institute for Regenerative Medicine. Here, I just want to highlight tonight that CIRM really has relaunched. And thanks again to all of the people who really helped us as a center and helped Californians for Cures across the state in order to make this happen. We were so excited in November when Prop 14 was approved by the California voters, and that relaunch is really happening. And so I just wanted to highlight that briefly tonight. Of the many things that we were anticipating relaunch of, including training internships, fellowships for graduate students, for postdocs, for medical students, shared resource laboratories, which we will be able to renew funding for, and of course the alpha stem cell clinics, discovery grants, and translational grants. These are all actually kicking off as we speak. In fact, there's a deadline for the Bridges program for undergraduate students that is just happening. We've been partnering with the Cal States locally, Cal State Fullerton, Cal State Long Beach, and San Bernardino in order to make that happen. And our first deadline for these big requests for applications from CIRM actually is already looming June 10th, when we'll be putting in a training grant application in order to refund those fellowships that we haven't had for the last couple of years. So we're super excited to have this off the ground, and we're working hard to make it a success. How can you help us do that? You can participate, sign up to follow our newsletter, follow us on Facebook, go to our YouTube channel and watch our previous videos. Be here tonight, which is fantastic to have the community involved and just listen to what there is to know about stem cells and how important we think that is. And of course, make a gift. We're really dependent upon partnerships to be successful, even for the training grants, for example, that we're putting in right now. We have more strength in terms of our applications if we're able to match those with internal funds. So that's a huge benefit to us. Again, thank you so much for being here. I just want to highlight the next couple of talks that are coming up. You are at this evening with Mike Hicks and Tahseen Mozaffar talking about muscle wasting and muscle disease. Our next and last of this current series is about breast cancer and a transcriptome of cancer stem cells in organs like the breast. And that's going to be Devin Larson and Kai Kessenbrock on June 8th. So after that, we'll have a bit of a hiatus for the summer, coming back in September, talking about brain organoids. So there's just laying out what our schedule is coming up. And with that, I just want to say a couple of words and introduce our speakers tonight. It is my great pleasure to present Michael Hicks, who is our one of our newest faculty recruits to the Stem Cell Research Center, just hired, honest to God, in the middle of COVID and starting up his lab during this period. He's an assistant professor in the School of Medicine at UCI in physiology and biophysics. He obtained his PhD in 2015 from University of Arizona, and after that did a postdoc in a fantastic spot looking at muscular dystrophies at University of California, Los Angeles. He's received a number of awards already in his short career. These include the 2020 Sidney Feingold Award, a 2019 Limb Foundation New Investigator Award, and the 2018 UCLA Award for Excellence in Postdoctoral Research. Our second speaker tonight is Dr. Tahseen Mozaffar, who has been a wonderful collaborator at the Stem Cell Center for many years now. He's a professor of neurology in UCI School of Medicine, of orthopedic surgery as well, director of the Division of Neuromuscular Diseases, and director of the UC Irvine ALS and Neuromuscular Center. He has a variety of awards, which I'm not going to tell you all about. Suffice to say that he's been honored many times for his teaching, particularly for the medical students and resident students. He's a past president of the Orange County Neurological Society. He has served as the Stanley Vandernoort Endowed Chair in Neurology at UCI, and is listed in multiple sites as one of the top physicians in the country as a neurologist. So with that, I'm super pleased to invite you to listen to what they have to say to you. I just want to highlight a couple of words in terms of format. In the interest of time and just some of the challenges that we face with doing these things virtually, I'm going to ask everyone to hold their questions to the end. You can put them into the Q&A. We'll answer them live if we can, or I will be compiling them and moderate a discussion at the end with our speakers. So our speaker presentations will be followed by a time for questions at the end. And with that, I'm going to turn it over to our speakers for the evening, stop my share, and invite our first speaker to go ahead and start theirs.
Thank you very much, Eileen, and everyone for attending the community lecture series today. I'm very excited to share some of the work that I've been starting here at UC Irvine and part of the work that Tahseen, Dr. Mozaffar, and I have begun. I'll begin the presentation with an overview of skeletal muscle and why it's a really interesting organ to study and why the stem cells of skeletal muscle are important. This will be followed by Dr. Mozaffar, who will talk about different muscle diseases, including diseases that he treats in the clinic. And then we'll talk about some of the exciting research that's going on here at the center. So I always thought if you're going to start with introducing skeletal muscle, why not start with a really fun video showing skeletal muscle? So I hope you all enjoy.
Skeletal muscles are responsible for the voluntary movement of the body. They are composed of fascicles containing multiple muscle fibers where each represents a single cell. Individual myofibers are covered by a basement membrane, which is continuous with the endomysium connective tissue layer. Peeling away the basement membrane reveals myofibers and the skeletal muscle stem cells, also known as satellite cells, which play a critical role in muscle fiber repair. Peeling away the sarcolemma membrane of the myofiber reveals the intracellular components, including myonuclei, that originate from the fusion of hundreds of myoblasts. The myosin and actin contractile apparatus gives us the ability to move in a controlled manner.
So thanks, I hope you enjoyed that. So my lab studies making skeletal muscle from pluripotent stem cells, and pluripotent stem cells can make any cell type in the body. But as you make skeletal muscle, you really need to be able to follow the developmental cues and how muscle develops in humans. And so part of my research and interest is involved in how does skeletal muscle form in the first place. So the body develops in three different germ layers: the ectoderm, endoderm, and mesoderm, and skeletal muscle is derived from the mesoderm. And as you could think, meso is metal. So as the mesoderm develops, it forms what is known as a somite. And somites form the musculoskeletal tissues. Those include your bone, cartilage, tendons, ligaments, parts of your dermamyotome, and your skeletal muscle. Skeletal muscle cells delaminate from the somite to form what we know as the limb and to establish different waves of skeletal muscle. So the primary wave of skeletal muscle established the first muscle fibers. And as these muscle fibers are developed, muscle progenitor cells bind to the sides of these muscle fibers and go through a secondary wave. And as they form on the sides of these muscle fibers, they mature and they become more functional over time. And as you could think about in fetal and in early birth, the skeletal muscle is rapidly dividing. And so when we're studying this from pluripotent cells, we can see all of the different types of morphology and fusion potential and cell matrix interaction that occur. And each one of these cells in development, such as the embryonic myoblasts, fetal myoblasts, and adult satellite cells, which are the cells that are responsible for muscle regeneration, can be uniquely identified in their proliferative potential and in their transcription factors, as well as in their ability to form niches. And so when we study skeletal muscle that's derived from pluripotent cells, we use development to benchmark the ability of skeletal muscle from pluripotent cells for things like disease modeling, right? So you want to get a cell that's more mature, similar to an adult cell, in order to really truly recapitulate things that are available in disease. And so when we think about skeletal muscle, we really think about its main function, which is to allow us to move. So skeletal muscle not only allows us to move, but it's involved in our posture and being able to stand up straight. The diaphragm controls our breathing, and skeletal muscle also controls our ability to metabolize. So you can think of things like diabetes, where skeletal muscle becomes really important. Skeletal muscle is a really unique tissue in that it's multinucleated. So if you think back to your biology class and you think about a nucleus within a cell, that nucleus controls things like your DNA and your genes. In a skeletal muscle, it's multinucleated. You can have 500 or even 1,000 nuclei inside of a skeletal muscle, and it's a very unique cell type in that regard. And that's because the skeletal muscle needs to pump out lots of proteins that allow for you to contract. And those contractile proteins are found within the sarcomere. So the sarcomere is your actin and your myosins, and those allow for contraction. And skeletal muscle is innervated by your motor neurons. And so when a motor neuron sends a signal to the skeletal muscle that causes an action potential. And so skeletal muscle is an excitable tissue, just like neurons, in that action potentials can travel throughout the muscle fiber and allow it to contract. There's also different types of skeletal muscles. So the muscles in your bicep are very much different than the muscles found in your gastrocnemius. And these are called your slow twitch and your fast twitch skeletal muscles. So your slow twitch are your more for long distance. There's your endurance muscles. And your fast twitch are more of your strength muscles. And these type of, in the lab, we can identify different types of skeletal muscles by things like which myosins they're expressing. And this is also really critical for diseases because your slow twitch myofibers in many cases are more resistant to disease. And so in diseases like muscular dystrophies, the slow twitch will be the ones that are longer survive versus your fast twitch muscles, which go down over time. And also that happens with aging. So that's an example of this graph here. It uses different types of myosins that can be marked, shown here in purple, that after 12 months of power training, and then DT stands for detraining, you can see that the type 1 myofibers are more resistant to the lack of training and exercise, and that's quantified here. And so understanding not only making skeletal muscle, but what types of skeletal muscle you're making become really critical for understanding how to improve function. The skeletal muscle is also innervated. So every muscle is innervated by one nerve, by one motor neuron, but one motor neuron can innervate several skeletal muscle. And the more skeletal muscle that are innervated by a single neuron can regulate strength. So if there's one nerve that has multiple muscles, those make for stronger muscle. And there's a number of really beautiful markers shown here. So this is the postsynaptic membrane is marked by the acetylcholine receptor. And then you have a presynaptic membrane from the motor neuron that innervates with this. And here at UC Irvine, we study a number of different neuromuscular diseases that are present. And so I'll talk about those in a couple of slides. The other thing that I'll talk to you about is skeletal muscle regeneration. And this is mediated by an endogenous muscle stem cell called the satellite cell. And skeletal muscle is one of the most regenerative tissues inside of your body. It's up there with your skin and your intestine and your blood that allow for muscle regeneration. And so for muscle regeneration, I thought it would be fun to introduce another video for you.
A major trauma to the muscle can be induced by massive injury, including toxins such as a snake bite. Even after extensive damage, small groups of healthy cells can be found. Following necrosis of the muscle tissue, the remaining viable cells, including muscle stem cells, engage in the regeneration process. The satellite cells activate and generate daughter myoblasts that begin the process of rebuilding the destroyed muscle fibers. During this period, residual debris is removed by the macrophages. The new muscle fibers are generated within the collagen matrix of the pre-existing basement membranes, following the differentiation and fusion of hundreds of myoblasts. Notably, during this regeneration process, satellite cells will self-renew and they are then set aside for future repair events.
So I hope you enjoyed that. So regeneration really lies on this dynamic interplay between the muscle stem cell, they're called satellite cells, and the stem cell niche. So as cells get disrupted from their stem cell niche, they activate and they proliferate. And I'll show that on the next slide on exactly what I'm referring to here. So, so skeletal muscle can remake an exact copy of itself. So upon injury, skeletal muscle becomes damaged and there's a transient inflammation of neutrophils that come and remove that debris and tissue. And then skeletal muscle begins to rebuild itself. And this is directed from the muscle stem cells. And over time, the skeletal muscle can make an exact copy of itself. And I believe that we've all experienced muscle damage at some level. This occurs when you even go and exercise. This occurs on a micro scale. But even at major injuries, we can regenerate the tissue. And this is, again, derived from the muscle stem cells that allow for this to occur. In chronic injury, such as in Duchenne muscular dystrophy, one of the diseases that we study, the skeletal muscle is continuously degenerated. And satellite cells, or the muscle stem cells, do their best to just repair that tissue over time. But other cell types, such as the inflammatory system and fibrotic cells, continue to accumulate over time. And they create physical barriers that block the muscle stem cells from being able to repair that tissue and reach each other. And they also send out different types of signaling that can disrupt the ability of the muscle stem cells to orchestrate regeneration. So muscle stem cells are marked by a transcription factor known as PAX7, and they reside on the side of the muscle fiber. So they're called satellite cells because you could think of a satellite cell orbiting the Earth. The satellite cells of the skeletal muscle orbit the side of the muscle fiber. And so upon damage, these satellite cells can repair an entire muscle fiber, which can have 500 to 1,000 nuclei within it. Only a handful of these, 5 to 7 of them, can repair that many nuclei. So they proliferate, they activate, and they turn on some other genetic markers such as MyoD. And these cells will make, several hundred of themselves. Many of those cells will then commit to differentiate. So they'll start to express other transcription factors like MyoG, and they'll fuse to make the first muscle fibers. And this is very similar to the process that occurs in development. The regeneration process transiently hijacks many of the same genes that you would find in development, and we're able to use those with pluripotent cells. And as the muscle fibers differentiate, a few of the cells take up position within the satellite cell niche and return to a quiescent satellite cell in a process called self-renewal. And this can occur over and over and over again for 100 years. And even in a centurion, you have quiescent satellite cells that are able to repair tissue. And so understanding this process of self-renewal becomes really important to understanding the regeneration process. And so this was a really fun paper that was published recently using a microscopy technique called intravital microscopy. And that's where you have a very high-powered microscope that can visualize inside of a living mouse. And this mouse had their Pac7 cells tagged with a yellow fluorescent protein, which is shown here. And it showed that as the skeletal muscle fiber died upon injury, these cells used the architecture of the skeletal muscle, the fibers, to proliferate and migrate along the side of this. And this is a live video of the satellite cells within the tube of the skeletal muscle proliferating and starting to fuse together to form new muscle fibers. And so it's very fascinating that these cells can hold on to the side of the extracellular matrix and fuse to repair damaged muscle fibers. Skeletal muscle regeneration does not occur independently of just the satellite cells. There are a number of other cell types within your skeletal muscle. You could think of your blood vessel cells, your capillaries. Those cell types are really your endothelial cells. You have fibroblast cells that create extracellular matrix, and you have other types of inflammatory cells such as your macrophages. And so number of labs have done very detailed studies looking at these cell populations over time following an injury. And they really occur in waves over time. And so you can see that initially, immediately upon injury, skeletal muscle becomes infiltrated by neutrophil cells that come in and clear debris. And then satellite cells become activated over time. But every one of these cells, including your fibroblasts and macrophages, go in these waves. And it's a very well orchestrated event for muscle regeneration. This becomes really important for diseases like the ones that Dr. Mozaffar will tell you about, where this regeneration process becomes disrupted, and other cell types such as your macrophages become chronically made, and they don't resolve. And then this can cause a number of horrible diseases. So when we're thinking about the muscles, skeletal muscle and the satellite cell, we could think about it on a number of different levels, and that is how the satellite cell directly interfaces with the skeletal muscle to form an immediate cell niche. And then we could think about this within the milieu of the different types of cells that are present, such as your motor neurons and your fibroblasts and macrophages. And then you can think of it much more on the systemic level on how satellite cells are interfacing with the other tissues, such as your bone, even all the way, things like with your liver and other cell types within the entire body to regulate muscle performance and overall homeostasis of the body. With that introduction, I would like to turn the slide over to Dr. Mozaffar to introduce more on some of the diseases that are studied here at UCI.
Thank you, Michael. It's my pleasure to build on the basics that Michael has talked about. And as Michael said, I'm a practicing neurologist, and I deal with a number of diseases that are related to issues germane to the topic that we are talking about, which is either muscle inflammation, lack of regeneration, a dystrophic process that may initiate the muscle process as well. I would say that Lou Gehrig's, who was a famous Yankee player, is the poster child for muscle atrophy. in some sense. And it maybe not as prominent in this particular photograph, but if you see the video of his farewell speech at the Yankee Stadium, you can see a very prominent atrophy of his hand muscles that was related to his disease, ALS, which is now known in the US as Lou Gehrig's disease. But Lou Gehrig was not the only poster child. There have been other diseases and other celebrities who've been affected by this disease. Audrey Hepburn had ovarian cancer, and towards the end of her life had very severe muscle atrophy that you can see in the picture on the top left. Rock Hudson, who was a handsome and very strong man, really wasted away to a skeleton of himself from the complications of HIV and AIDS. And then there are diseases that I deal with in the clinic, myotonic dystrophy, which is a genetically inherited muscular dystrophy, which has very prominent muscle atrophy. And you can see in this gentleman, very severe atrophy of the face muscles, the temporalis muscle, as well as the muscles around the neck. ALS we talked about, and one of the hallmarks of ALS is the atrophy of the intrinsic muscles of the hand, and oftentimes that's how the disease starts. Charcot-Marie-Tooth disease, which is another genetically inherited neuromuscular condition where you have these skinny legs because your muscles are atrophying away, and that because of lack of nerve input. But one particular disease that, and as I get older and the population at large keeps getting older, one of the diseases that everyone sort of talks about and fears is a disease called sarcopenia, which is a disease, age-related atrophy of the muscle. And it's an area of intense research at this point. So muscle atrophy can occur in specific muscles. So if you cut a nerve, only the muscles affected by that nerve will be undergoing atrophy. It can also happen with inactivity. So one of the challenges with space flight, especially missions to Mars, is it will take 3 months to get to Mars. And by the time the astronaut get to Mars, their muscles will have atrophied by about 20% because of lack of activity and the loss of gravity on that. It can be a systemic response to fasting or Aries disease. So kidney disease, for instance, especially patients who are on hemodialysis undergo muscle wasting. As I already mentioned, cancer and HIV as being two of the more notorious diseases that cause muscle atrophy. And during atrophy, there is a progressive, pervasive process that leads to degradation of the myofibrillar apparatus, which Michael showed, which is responsible for contraction of the muscle. And there is acceleration of the degradation of the soluble proteins. In systemic illnesses, unfortunately, your protein synthesis is also decreased. So think of muscle as pure protein. So when you lose muscle protein, one of the ways to balance that would be to have more production of protein. So you have a net negative, you have a net balanced process. But because you have reduced protein synthesis and you have an accelerated protein degradation, you will have muscle atrophy. Muscle atrophy does cause significant disability, and in some cases, it may cause mortality as well. Cachexia is a term that we use for severe loss of body weight, especially muscle mass, with or without loss of muscle fat. Cachexia is associated with serious diseases, especially the term is often used synonymously with cancer. But atrophy and cachexia are mediated by induction of specific genes that are responsible for muscle breakdown, and these genes are collectively known as atrogenes. So the muscle atrophy paradigms that one can study in a clinical model or a research site is age-related sarcopenia, and this is the holy grail because we want to make sure that we stay fit even as we old. Physiological responses to fasting and malnutrition, muscle atrophy during systemic illness or cancer-associated cachexia. If somebody stays in the ICU for too long, they can get muscle atrophy just from the disuse, but also from some of the other diseases that they may have during that period. As I mentioned, in patients who are undergoing renal dialysis, cachexia becomes a huge issue. There's also a muscle loss that happens with patients with COPD. And then there are neurological conditions that do that as well. Sarcopenia is not only affects muscle, but it actually also affects bone. So it is a systemic atrophic process of the muscle and bone. These are seen in patients who otherwise don't have any disease that may be responsible for this kind of muscle loss. And we estimate that it affects as much as 15% of the population over the age of 65 and up to 50% of individuals over the age of 80. Now, this is what sort of sarcopenia would look like. So there is a diffuse, not focal, but diffuse atrophy of the muscle mass. So this is what normal muscle looks like. This is what a loss of muscle in a sarcopenic patient looks like. And you can see that also on MRIs, where you can see a very full MRI with muscle, and you have significant and qualitative loss of muscle over time. So we know what causes atrophy. Atrophy is caused by a balance or imbalance in the positive genes as well as the negative genes. So a lot of factors cause muscle atrophy. You have catabolic states, so burn or uremia related to kidney disease or cardiac disease will induce some of these genes, such as myostatin, and will also inhibit or reduce some of the more positive genes, such as insulin-like growth factor. If you don't exercise, there is a consequence of that. If there is inflammation or there is diseases like HIV, that also kicks off a bunch of inflammatory markers. And all of these eventually cause problems with either the myostatin pathway, which are the SMAD2 and SMAD3s, or the IGF signaling pathways, which are the AKTs and mTORs, and the NF-kappa beta. And eventually they all go into the nucleus and turn off the production of the muscle genes or the muscle transcription factors that lead to production of new muscle. So this is a simple way of looking at it. This is the IGF-1 pathway, where IGF-1 pathway's main role is to inhibit the production of atrogen 1 and other atrogens through activation of something called FOXO-P. But in diseases that are responsible for atrophy, the IGF-1 pathway is blocked off. There's less production of AKT, there's less phosphorylation of AKT, the FOXO protein is no longer phosphorylated, and you get upregulation of these atrogens that eventually lead to protein degradation. Muscle is an important apparatus in the system. It is responsible for muscle contraction. It helps us move. And when you have inactivity or disuse, then you get breakdown of this. And there are specific proteins, which we call proteases or ubiquitin ligases, that are responsible for breakdown of these. So normally, as Dr. Hicks showed you, there are these thick filaments or myosin filaments which attach to the actin filaments, and they are responsible for muscle contraction. But they also have a built-in cleaning system, which is activated by the MRF1 or TREM32 pathways. And when you have atrophy, the MRF1 and TREM2 will dissociate the myosin from the actin, and then eventually it leads to breakdown of these. So there are a lot of the work that's going on in preventing muscle atrophy is understanding how we target the MRF1 and these different ligases that may be responsible for protein. So in the different disease paradigms, you have different or differential activation of some of these atrophy-inducing markers, whether it's the insulin resistance pathway, whether the increase of the glucocorticoid pathway or the myostatin pathway. So here I've listed a bunch of diseases and what goes up and goes down in this. And you have to understand this to be able to appropriately treat these patients. So one of the focus very recently in the pharmaceutical industry was inhibition of the myostatin pathway. And the myostatin pathway became important because in the lab studies or experimental studies, it was shown that if you inhibit the myostatin, you get these super mice, which have very, very well-built muscle. So myostatin is normally involved in regulation of muscle growth. It actually inhibits muscle growth. So when you knock out myosin or get rid of it, you get massive buildup of muscle. And this obviously is an induced mechanism, but it also occurs naturally. So there are these naturally occurring cattle. These are called the Belgian blue bulls or blue cattle, which have massive hypertrophy or increases in muscle mass. And these animals actually carry a naturally occurring spontaneous mutation of the myostatin. So their muscles grow to a much larger size on that. And about 10, 12 years ago, there was a child born in Germany who even at birth was very muscular. So you can see these pictures from this New England Journal of Medicine paper, where you could see that even at neonatal age, very early on, had fairly large muscles that persisted beyond the neonatal period. And this is this child at the age of 6, and he's buffed, he has very well-formed muscles, and he can bench press more than, way more than what the kids at his age can do. And this child also has a naturally occurring mutation in the myostatin. In fact, his family has this mutation, and they all tend to be very muscular. And that's raised the concern that maybe some of our more gifted athletes who have built muscles may have variations in their genes for the various muscle genes that regulate muscle size. So in some ways, some kind of a natural gene doping concept evolves out of that. IGF-1 is the other pathway. So myostatin is more of a negative pathway. It reduces muscle size, whereas IGF-1, if you give IGF-1 to an animal muscle, it grows the muscle size on that. So these are, again, animals that are IGF. These are the wild types. And then you have mice that have overexpression of IGF, and you can see increases in muscle mass compared to the wild type. That is very common. And IGF-1 has been targeted in a lot of diseases, but unfortunately, one of the challenges with IGF-1, this IGF-1 also is involved in cancer formation, oncogenic processes. So you have to be careful with IGF-1 because you may induce a bunch of cancer genes that you do not want to induce as part of it. One of the diseases that I focus on is inclusion body myositis. And I think it's a perfect example of a combination of all these factors that causes the muscle atrophy. On that. So inclusion body myositis is the commonest muscle disease in adults over the age of 45 years. And the more you look for it, the more you find. And my fear and concern is that a lot of older folks, when they have muscle weakness, we chalk it off to their age-related sarcopenia. But if you carefully look at them, they may also have inclusion body myositis. And we've seen a fair number of these examples. So the concern is not overly out there. I think it may be a real, it is a slowly progressive painless disease, but it becomes very disabling with significant disability with simple things that are relevant to daily activities. And it's a very prominent pattern. It's a proximal weakness in the legs, mainly affecting the hip and the quadriceps muscles or the thigh muscles, but very prominent distal muscles. So you could get to a situation where your finger muscles are weaker than your deltoid muscles. It is a predominantly male disease, although you can have female involvement. And there is very prominent involvement of the bulbar muscles, which are the muscles of swallowing and chewing. But also, the more I study this and the more amazed I get is how involved is the face muscle as well, especially towards the advanced stages of the disease. And then there is reduced life expectancy Most of it is thought to be related to involvement of the bulbar muscles, but they may also be a respiratory muscle involvement there. And these are some of the features. So I'd like to show this picture because one of the ways to diagnose this is just ask the patient to make a fist. This is a diagnosis you can make from the door. You don't even have to examine the patient, but obviously we like to examine the patient even more. So it's an asymmetric disease. So one side is more affected than the other. And you can see that whereas he can make a fist on the right, on the left, he cannot bury his fingers because these fingers are weaker. There is asymmetric and prominent atrophy of the quadriceps muscle. So you can see that this quadriceps muscle has scalloping or atrophy. There's also a similar atrophy of the forearm flexors, which you can see very prominently on the MRI. And we use a lot of muscle MRI because sometimes the muscle MRI shows us the changes way before the clinical exam shows. Here is an MRI of the thigh muscles, and you can see that it's predominantly the quadriceps or the anterior muscles that get affected. The hamstrings, which are the back muscles, are relatively spared early in the disease. But even within the quadriceps, there is this very peculiar sparing of the rectus femoris relative to the vastus and other diseases, other muscles there. The pathology is very unique. And it, again, as I mentioned, there's a perfect combination of muscle atrophy, which is related somehow to inflammation. There's a very prominent lymphocytic infiltration that goes on with invasion of non-necrotic fibers. There are these degenerative processes as well. So you have rimmed vacuoles, which are present, and these are usually nuclear debris. You have a lot of muscle atrophy, but also a fair amount of muscle regeneration. You have mitochondrial abnormalities. So the mitochondria in these muscles are not normal. They have large deletion. So you see a lot of mitochondria that are missing essential enzymes such as cytochrome oxidase. And you have buildup of these abnormal proteins in the muscle, such as in this particular example, it's P62, but you can similarly see TDP43, you can see SMI31, you can see amyloid, all sorts of other diseases. And then on ultrastructure and electron microscopy, you see these tubulo-reticular inclusion, and that's how the disease got its name of inclusion body myositis. But you can see a lot of inflammation. So these are different inflammatory markers, CD8, CD68, that will show up. There is a fairly diffuse and massive upregulation of MSC class 1. And this is the evidence that suggests that there is a autoinflammation or inflammation may be a primary issue here in this disease. And then this is an example of TDP43 expression in the muscle that we routinely stain for when we make a diagnosis of things. There is muscle atrophy, and there is very good data to suggest that the muscle atrophy is related to the inflammation in the muscle. So in an experimental models, when you reduce the inflammation, you actually allow the muscles to grow back and regenerate. And then working with Dr. Vialta, we have been doing a fair amount of work and characterizing the inflammation in the blood. And we plan to do the same things in muscle. And we have shown that it's mainly a lymphocyte or CD8 driven disease. But even within the CD8, most of the inflammation comprises of these highly differentiated, what now called as senescent lymphocytes. So these are overstimulated, highly differentiated cells that now lose their capacity to divide or undergo apoptosis, which is the natural cell death, and then become resistant to all sorts of conventional therapies. This particular graph shows that if you look at these CD8 cells and you try to stage them, it's predominantly the highly differentiated cells that become that are responsible for the inflammation in IBM. So this is an area that we want to research further, and we have a nice collaboration going with Dr. Vialta on this, as well as with Dr. Hicks on that. So again, just to summarize, inclusion body myositis is very slowly progressive disease. there's an increased mortality and morbidity and disability associated with disease, but there's also an increased mortality. And for the last 20 years, there have been multiple attempts to treat the disease, but the disease remains untreated. We had multiple clinical trials, including two recent trials, one by Novartis and one by Offazine, that spent a lot of money on these trials and completely failed on that. So part of the challenge is that there are lots of unknowns in this disease. We really don't fully understand the role of these very prominent inflammatory infiltrates in this muscle in terms of disease pathogenesis. We don't understand the natural history. There have not been enough studies studying the disease over time. Especially, there are now well-defined biomarkers such as antibodies as well as these variant lymphocytes. So we really would need to understand how the disease behaves. So if you have an individual who has an antibody but against an individual who doesn't have the antibody, is the disease different? Is the disease behavior different? Are they going to respond much more to treatment or not? Some of the outcome measures that we use in clinical trials may not be well suited to this disease. So one of the focus that we would like to have on is to study the optimal outcome measures. Right now, the outcome measures are very crude, but we are hoping that we can use some imaging-based or other more sophisticated outcome measures. Bulbar insufficiency, which leads to problems with swallowing and maintaining their weight, is a major contributor to disease mortality, but has generally been ignored and not very well studied. And our biggest challenge is that we don't have a validated or good animal model for IBM. Dr. Frank LaForla many years ago had created a animal model that recapitulates some of the features of the disease, but not all the features. That particular animal model didn't have much in the way of inflammation. So that's been one of the limitations, but there are some new advances that I will mention later on when we talk about some of the other grants that we have received. So at this point, I'm going to hand it back to Dr. Hicks for him to continue with his talk.
Thank you, Dr. Mozaffar. So I'll tell you, so the lack of a good mouse model is a good segue into some of the research that's going on here at the center. And what we do have are the ability to make human models in a dish. So we can take patient sample biopsies from the skin of patients, for example, with inclusion body myositis. And we can reprogram the cells taken from the patient and make induced pluripotent stem cells. And so induced pluripotent stem cells are able to make any cell type in the body, and that includes skeletal muscle that we can use for preclinical screening and disease tools. So my research is focused on a different muscular disease, which is called Duchenne muscular dystrophy. But Dr. Mozaffar and I have been in discussions about developing a biobank from patients that have inclusion body myositis, and then trying to understand what are the genetic defects that occur in those cells, and then using the skeletal muscle in a dish to screen for new drugs that could potentially improve some of these clinical trials. So these are the skeletal muscles that we're able to make in a dish from iPSCs. And so as Dr. Mozaffar had mentioned, the inclusion of IGF-1 and the inhibition of myostatin can make skeletal muscle, and that allows us, and those are precisely some of the signaling pathways that we target to improve skeletal muscle differentiation. So this drug right here that we use in a dish targets the myostatin pathway and inhibits it. So you can see in a dish, when you inhibit myostatin, you make much larger skeletal muscles than without that inhibitor. And so following some of the biological and developmental paradigms, we're able to make better skeletal muscle in a dish for disease modeling. And so the disease I'll tell you about as an example is Duchenne muscular dystrophy, which is a progressive muscle wasting that causes premature death. It's an X-linked disease, so it affects boys, and there's no cure for this disease. Duchenne muscular dystrophy affects dystrophin, which is shown here. It links the cytoskeleton to the extracellular membrane of the muscle fiber, and without dystrophin, the muscle, when it contracts, it breaks apart and dies. So what we've done is take DMD patient fibroblasts, and using CRISPR-Cas9 editing, we've been able to remove the missense mutation that occurs in Duchenne muscular dystrophy and make a shorter truncated protein that then restores dystrophin and makes a much milder disease called Becker's muscular dystrophy. And people can live with Becker's for a very long period of time. And so we've taken the CRISPR-Cas9 edited lines and derived skeletal muscle cells, and then looked at their ability to make skeletal muscle in vivo as a cell replacement therapy to restore dystrophin, and also to replace the diseased satellite cells with healthy new satellite cells so that you can continuously make new muscle over time. And this is what this looks like when we do the CRISPR-Cas9. We differentiate our cells. You can see that the DMD iPSC skeletal muscle, they express myosin, but they lack dystrophin, whereas the CRISPR-corrected DMD iPSCs, they re-express dystrophin. And we can disease model this in a dish using here called the creatine kinase assay. And so you change the osmolarity of the media that these myotubes are grown in, and as you decrease the osmolarity, the cells become stressed and they will release creatine kinase into a dish. So you can see in wild-type derived muscle fibers are pretty resistant to this osmolarity stress. But if you don't have dystrophin, the cells will bust apart, release creatine kinase all over the place, which causes muscle death. And we can see with the corrected lines that we can actually restore muscle function and survivability. So this is one example of how we've done this. We've also shown this in transplantations in vivo. So in this case, we've transplanted in muscle derived from these iPSCs, and then we cardiotoxin injure a mouse model of muscular dystrophy. It's immunocompromised, so it can take the human cells. And after 30 days, we stained for human dystrophin. Wild type cells express the human dystrophin. The DMD iPSCs don't. Well, whereas our corrected cells can then restore the dystrophin missing protein. Dystrophin serves to anchor the sarcolemic membrane, the dystroglycan complex, to the muscle membrane. Wild type cells show the dystroglycan complex is shown here with the beta dystroglycan. The DMD cells don't, whereas our cells are able to restore functionality and new proteins that were once missing to the muscle membrane. So we can do this with different iPSCs, and not just for Duchenne, but for a number of different diseases. We've continued to improve our directed differentiation protocol. So when you make muscle, or when you make any cell from a pluripotent cell in a dish, there's other cell types that are formed, and we can enrich for the skeletal muscle cells specifically using the flow cytometry core here at the stem cell center, and we can get a very enriched population. And so we can see that the skeletal muscles that we transplant can form these multinucleated muscle fibers, and we're transplanting into a mouse in this case. So it forms a multinucleated muscle fiber that fuses with the endogenous mouse cells, but also will form these human-only myofiber regions. And this provides a unique opportunity to understand how muscle is made. So these newly formed de novo muscle fibers need to innervate. They need to form tendons. And we can study what are the factors that regulate tendon formation? What are the factors that regulate neuromuscular formation with this new model? It's a humanized mouse model. It's a mouse that has now human cells in it. And we can tell that they're human-only cells because if you take a longitudinal section, you can see here in red are the human cells, and every single cell within these multinucleated muscle fibers are of human origin. So this is what this looks like, and it allows us to explore the possibilities of what do you do now with a humanized mouse that has skeletal muscle in it. So as I mentioned, in the human-only side, we can study how do de novo muscles form. How do stem cell niches form? can we use this information to improve regeneration and aging and things like developmental defects when skeletal muscle forms for the first time? We also want to know, how do these cells integrate with other tissues, such as the nerves? And these chimeric regions, we can use these to study the restoration of missing proteins, or look at inclusion bodies, such as TDP43, an inclusion body. So rather than have a mouse model for IBM, we now have a human model that's inside of a mouse. And we can study a humanized mouse for drug screening and a number of different diseases. And this can also be used for studying how muscles innervate with motor neurons, such as ALS or spinal muscular atrophy. And so pluripotent stem cells really have the unique advantage that you can genetically engineer a pluripotent stem cell to express any protein. In this case, we used a red fluorescent protein. It was inserted into a safe harbor locus, which has no disease or cancer-causing ability inside these cells, and it remains pluripotent. So you can genetically engineer your cell to do basically whatever you want it to do, and then differentiate it into the skeletal muscle. And this is also possible here at the Sue and Bill Gross Stem Cell Center because they have a CRISPR-Cas9 core. And so instead of just developing reporters, you can use CRISPR-Cas9 to inhibit or activate any gene of interest and do screens. And you can study things like inducible stem cell niche formation or inducible ability for the innervation of your cells. How do you improve contraction? And we can do this, like I said, here with many of the valuable philanthropy that's available that help to fund the CRISPR-Cas9 core here. And this has been really a big help and able to get research for understanding what is the genetic causes of diseases in skeletal muscle. So I'll give you one example of how we're using that core here in my research. So last year, I co-published a paper that built a microfluidic device that had skeletal muscle on one side and motor neurons on another side. And those motor neurons would have axons that would reach through a microfluidic device, and they would innervate that skeletal muscle. And so the skeletal muscle would actually send signals to the motor neurons that would allow for the axons to be guided through these microfluidic channels and bind to the muscle neuron. And there's this really interesting ability of the rabies virus. So you think about getting bit by a rabid animal. It bites you in the muscle, and it sends a virus from the muscle into the motor neuron, into your central nervous system, and that's what causes the disease. So in this case, we engineered a rabies virus to have a red fluorescent M-cherry protein, that's shown here. And over 7 days, we watched the red fluorescent protein travel from the skeletal muscle into the motor neuron. And this is a process called retrograde signaling. And retrograde signaling from the skeletal muscle to the motor neuron could enable different ability. So instead of sending a virus, why not send a neurotrophic factor that improves neuromuscular formation and survival of the motor neuron for diseases such as ALS or denervation. And so that's shown here. We have the skeletal muscle that's red, and then after 7 days, we can see that the motor neurons are actually turning red because of their retrograde signaling that occurs from the muscle neuron. And so I've shown you that we can make these human skeletal muscle inside of a mouse. And shown here is acetylcholine receptor, which is the postsynaptic membrane of the skeletal muscle. And these are not innervated by the mouse, and they need to be able to send a signal from the skeletal muscle to the motor neurons, a come to me signal, and innervate. And you can think of the clinical applications for this for all of the different neuromuscular diseases. So in combination with the CRISPR-Cas9 core, I want to engineer neurotrophic genes into the 3' region of muscle-specific genes. So when the skeletal muscle differentiates and becomes a muscle, it will turn on a muscle-specific gene. And then in that region, it will now start to secrete neurotrophic factors, your favorite ones like BDNF or GDNF, and that can influence skeletal muscle, neuromuscular attraction to the skeletal muscle, and it allows for an entire new way of thinking about how does muscles and nerves innervate with each other. And with that, I would like to turn it back over to Dr. Mozaffar to complete the last couple of slides about some of his really interesting new NIH-funded studies. If you want, I can flip the slides for you.
Yeah, thank you, Michael. So we had the honor of being selected as one of the lead site for a 5-year study that is going to address some of the shortcomings that I mentioned in terms of IBM research. So a lot of the unmet needs that I talked about. So what this NIH funding will allow us to do is to create a 13-site consortium across the US, where we will bring in these patients and do some of these studies of natural history, as well as looking at their blood biomarkers. We are also planning to do a muscle study. And this will create a structure to be able to do future clinical trials, but also do a variety of other studies, because you will have the patients and the biospecimens to be able to do this. For the purposes of the, if I can have the next slide, Michael. For the purposes of this study, we stuck to the natural history study and defined a path where we could study the natural history of the disease and behavior in relation to 2 specific biomarkers. One was the anti-5C1A antibodies that are present in about 70% of these patients, but are very specific, but also in relation to these senescent immune cells that I talked about, and see if that influences the disease behavior in any way. But we also want to look at muscle biopsies and in relation to these biomarkers, and see if the muscle tissue behaves differently in the presence or absence of these markers. And this is a great opportunity because in addition to the CRISPR-Cas9 core that Michael was talking about, we also have a flow cytometry core, and we recently acquired the CyTOF machine. But attached to the CyTOF, we also just got a grant that will allow purchase of a machine called Hyperion, which allows us to look at multiple antibodies at the same time on a tissue section. And you can do all sorts of investigative studies to see what's happening in these muscles. And we also want to look at the respiratory system, which is involved, but has not been very well studied in IBM. Next. So there are other collaborations. Dr. Vialta and I are collaborating with Dr. Lloyd at Hopkins. So similar to what Dr. Hicks mentioned in terms of creating these mixed muscle models where you have part of the muscle belonging to the animal and part of it is humanized. There's a similar setup with mouse xenografts where you take human muscle biopsy samples and you put it in a nude mouse, which doesn't have any immune system, and you let it grow there. And eventually you have a part human, part mouse muscle, where you can study a lot of these things. And Dr. Vialta has been doing a lot of studies in collaboration with Dr. Hopkins. Dr. Vialta's lab is working with our group to further characterize the antigens where these immune cells are responding to. This is the holy grail in IBM. We know it's an immune disease. We really don't know what the immune cells are reacting to or reacting against. So we've been doing some single cell RNA sequencing studies in relation to this. And as Dr. Hicks mentioned, we are just starting to do some work with his group to create pluripotent stem cells, and then to further develop this work to understand the disease progress or disease pathogenesis. And then there's a lot of effort going on in terms of developing new drug trials, including a drug that was developed here at UCI called dilazatide, which specifically blocks a potassium channel called Kv1.3. But we've also been talking about senolytic therapies, which specifically target these aging senescent lymphocytes. We are working together. So Dr. Vialta, Dr. Hicks, and I are collaborating to create a muscle study group or a muscle biology group here at UCI, eventually with the hope that we can create a center for muscle research, looking at both basic science aspects of muscle, but also translational aspect, including clinical aspects of it. And there are a number of individuals on the UCI campus that have an interest in this on muscle studies, and we've already created this group. In addition, on the clinical side, our group holds annual neuromuscular colloquium, which is an incredibly academic meeting where we bring together people from all over the country that are practicing neuromuscular physicians as well as scientists to talk about relevant issues. There's also a pathology seminar that we run as well. So there is a strong collaboration here, a strong setup of muscle groups.
So thanks you guys very much for your time tonight and for such a broad, encompassing presentation. We do have a couple of questions that have come in from the audience. So if you could keep your video cameras on, I'll just walk through a couple of those and try and pitch them to the appropriate individual. The first one, I think, Tahseen, would be most appropriate for you. Are you aware of any drugs that are commonly used in the bodybuilding community that might be used for muscle repair and enlargement, and could those have clinical applications in some of these diseases? And just my quick message to the audience, I'm paraphrasing probably a couple of questions together, so I apologize if I don't get your exact question and I'm rolling it in with something else.
So
Tahseen, go ahead.
Yeah, thank you, Eileen. That's a great question. And so there are a number of drugs that have been used for this purpose. I mean, most of it are, as the person who asked the question mentioned, these anabolic steroids or androgen, which have androgenic properties. So they are derivatives of testosterone or the male hormones. And obviously, we know that they can grow muscle mass, muscle size. They can potentially make muscles stronger, but they have side effects, and especially concerns for liver toxicity, but also what we call virilization, which is they make you manly. Manly meaning that there's hair loss, there is deepening of the voice, which are probably okay characteristics in a male, but when even females use this, that's not a welcome trait on that. But there's also concerns for things like prostate cancer, because it may grow, prostate cancer may grow in an androgenic environment as well. As I mentioned, there has been an effort to develop drugs that are based on the IGF-1 pathway. Most of those trials unfortunately failed. And then the myostatin inhibitors, and a number of companies, including Novartis and Wyeth, have been involved with myostatin inhibitors. They do grow muscle size. So in all of these failed trials in ALS, as well as Charcot-Marie-Tooth disease, and the recent one in IBM, the muscle size did increase, but the muscle strength does not necessarily increase. So there's a disconnect where you can grow the muscle mass, but you don't necessarily increase the power of the muscle. In addition, we create creatine, which is a powder supplement that a lot of bodybuilders use, does increase muscle size. Some of the beta-2 agonists, such as albuterol, can increase muscle size, but again, those have significant side effects as well. So there is not a magical drug that is available, at least commercially, that may be the solution to this. I mean, there are boutique or custom drugs that are used by super athletes that may be failed to be detected by testing. And as I mentioned, there is a concern that there may be, at least in the athletic community, there may be gene doping going on, where are drugs given that enhance the function of some of these genes. So you know that some of the athletes use erythropoietin, which increases red cell quantity, but there may be other gene doping. There was a very nice article in Scientific American a few years ago that talked about this particular issue.
So I think maybe a related question to this, and you've touched on it a little bit, but for example, a genetic condition where there's, an overly robust muscle development. For example, the other side of that on a clinical perspective is there a negative impact of that? Are there late onset diseases or issues that may come, for example, from the kindred you showed, the child who was carrying a mutation that gave excessive muscle development? Are there potential problems that would come from, augmenting muscle in that way?
Yeah, we don't know that answer to that yet. So that child is, I think, 12 years old right now. To my knowledge, there are no issues right now, but we don't know what the effect of this prolonged stimulation of the muscle will have in the long run. So that's a great question, but I don't think we have the answer to that yet.
And I would also
sorry, go ahead, Mike.
On that. There are developmental studies where some birth defects are caused by precocious differentiation of the muscle stem cell pool. So if your muscle stem cells do not last into your adulthood and they differentiate too early, they kind of lose their shot, right? And the muscle forms in this embryonic state, and you end up with these birth defects, limb birth defects where people don't fully develop. So being able to keep the stem cells not too proliferative, but in this Goldilocks zone where they can continue to self-renew becomes really critical. And so that's the other side of it too.
So that's a great comment. And I think it sort of leads into a different question in terms of muscle regeneration and ways to augment muscle. Then, Mike, from your point of view, is it the stem cell pool that it's important for us to think about manipulating? Would you want to do muscle transplants? And what is the role of inflammation in that? If you're having ongoing inflammation, is some good, but a lot is bad in terms of those pools? Does it affect what their final development is? So we have a sort of a host of questions that span those things.
So the muscle stem cell is the primary driver of making new muscle, right? If you knock out the muscle stem cell, if you ablate those, you unequivocally block the ability to regenerate. So they become very critical. Interestingly, in some diseases, such as Duchenne, you just have dysfunctional muscle stem cells. They just don't grow correctly. They will divide, but they don't actually end up fusing correctly. So you can have non-functional or exhausted stem cells. And then on the other side, the stem cells receive their cues from the extracellular environment. And from the niche point of view, that's primarily the muscle fiber itself, but it's also the inflammatory cells. And the inflammatory cells come in waves. So they can lead to both the activation of the muscle stem cell as well as the dampening of the activation response. So those cells need to be able to return to a quiescent state. Otherwise, they enter this proliferative state where they're no longer able to repair future injuries. And so they become exhausted. So the inflammatory system, the fibroblast cells, the blood vessel cells, all play a critical interdynamic role in studying how the skeletal muscle stem cells function. And furthermore, the inflammatory cells can secrete extracellular matrix or induce the fibroblasts to secrete it, which causes physical barriers and scar tissue formation that the skeletal muscle itself can't burrow through. The skeletal muscle doesn't have this endogenous ability to just break all the scar tissue formation apart. And so all of these other cell types become really critical in understanding the regeneration process and also how regeneration goes awry in these muscle diseases.
So Eileen,
go ahead.
So in relation to, for instance, IBM, and I think I briefly mentioned this, but in the xenograft model that Tom Lloyd created, there is obviously very prominent inflammation, and there is clear evidence for lack of regeneration of the muscle. But if you irradiate that muscle or get rid of the inflammation, and they also did this recently with using an antibody called OKT3 antibodies, which is a drug that's used in humans, but they used it in this mouse model. When they eliminate the inflammation, the muscle started regenerating. So there is a negative influence of the inflammation on possibly the satellite cells or muscle regenerating cells. So again, that's something that really is interesting and curious in IBM. but I'm sure it also happens in other diseases as well.
Great. So I'm trying to decide which question to ask next. I think, Tahseen, since you just sort of ended on IBM, we have a couple of questions that come in on that. So let's go there. And one of them is whether there's a genetic contribution or risk, genes that would put you at risk for IBM, inclusion body myositis. And does the initial atrophy in certain muscles in IBM spread to other muscles? And I'll stop there and then I'll come back with two more once you've answered that.
Sure. So we generally don't think of IBM as a genetic disorder, but there are some risk genes that have been identified in IBM. So one of them is VCP, and there are bona fide cases of what we thought were IBM, and they behaved like regular IBM, who turned out to have mutations in the VCP gene that Dr. Kimonas had described many years ago on that. But similar to VCP, there are other, there's one other gene that is again related with protein degradation that has been identified as a potential risk factor for IBM. So there have been somewhat small gene association studies that have been done in IBM so far. Those are the two main genes that have been shown, but the role of there in terms of accelerating disease or defining disease has not been well identified. There is an effort right now. It's a multinational, multi-center collaborative effort that we have also contributed to through University College London, which is attempting to do a genome-wide association study in IBM to further define this as well. And then like any degenerative condition, so as we know in Alzheimer's and in Parkinson's and ALS, the disease is asymmetric, starts on one side and then spreads on to the other side. And there is this concept of regional spread where it goes from one arm, let's say, to the other arm. There's a similar spread that happens in IBM. So it will affect the other muscles as well. And there is a predictable pattern to and it sort of raises a question mechanistically of how disease propagates in an individual, and they may be cell-to-cell distribution or transport of these proteins that may be causing the degeneration. So there's been a lot of work going on studying that right now. But as far as the motor neurons are concerned, I'm not sure that anyone has fully studied what happens to the motor neurons that used to innervate these lost myofibers. There is a small degree of neurogenic involvement in IBM as well. But I have to say that the neurogenic side of it or the motor neuron side of it has not been very well studied.
So great answer and very comprehensive to what was asked in the Q&A. Going, I think, to the next question that's there, and I think you can probably both comment on this. It's a question about cardiac muscle, and this is, of course, coming from the perspective of someone who has a heart attack or an ischemic event that's affecting their cardiac muscle, obviously very common in the American population. And I mean, basically, the essence of this is why doesn't that muscle regenerate? Are there satellite cells? Are there not satellite cells in that cardiac muscle? Why can't the heart fix itself? And this has certainly been a very active area of research in terms of stem cell research with, you know, a variety of outcomes in terms of published studies over the last several years. So any comments that you guys might have on that? Mike, do you want to kick that off? And then we'll go to Tahseen next. Yeah.
This isn't true in every species and not true in development, but in the adult, you have mature cardiomyocytes, and they're not the same as a satellite cell. So they're not very proliferative. And so they're not able to regenerate the similar way that you would have in your skeletal muscle. Skeletal muscle is adapted to have these satellite cells because we experience injuries all the time. Whereas in the cardiomyocytes in your heart, they don't regenerate as well. People have been looking at this in different models because that's not the case with, say, like a zebrafish. Zebrafishes can regenerate their hearts fairly well. Early in development, we do have the machinery, like in mice, to be able to regenerate cardiomyocytes. But as they mature, they seem to lose this potential. There's some really good work out of the University of Washington ran by Chuck Murray that uses pluripotent-derived cells to make cardiomyocytes in a dish. And his goal is to get 1 billion cells, literally 1 billion cells, and put them into the rhesus macaque monkey. And you can see large areas of these embryonic cardiomyocytes derived from pluripotent cells in a dish to regenerate the heart. But yeah, the skeletal muscle and the cardiac muscle behave differently. It becomes important in a lot of these diseases, such as Duchenne muscular dystrophy, where dystrophin is also missing from the cardiac cells. And so the patient in general die from myocardial infarct from the heart parts. So it's important to think about, but they don't have the stem cell like muscle.
Yeah, I don't think I have much to add to that. I mean, that's been bigger a challenge is that whereas skeletal muscle is very adaptive and part of it is related to our ability to exercise, but also the frequent injuries, once the heart gets involved in an adult, it's a very difficult thing to repair. So I think the issues that Michael brought up are interesting issues.
So can I just expand on that for a second then? Because I think that was a really great comprehensive answer. Is that I mean, there are different sorts of stem cell trials that are ongoing for myocardial infarction. So for heart attacks, for ischemic events in the heart. Does that make sense? Actually, is that something you think is ready for prime time from a clinical perspective? And I don't know if Tahseen, maybe that might make the most sense for you, but Michael looks like he might have some opinions in terms of chiming in there.
Yeah, I think Michael may be a better person. I mean, it's the same thing. I mean, there are sort of efforts being made to do these kind of testing, but I'm not sure how successful they would be because of some of the inherent limitations. But Michael, I mean, I think I'd love to hear from you. What do you think?
So there's really 3 areas for the heart. You could think of deriving a pluripotent-derived cardiomyocyte, which still has some proliferative potential because it's an embryonic derived. There's other work that's going on that uses other types of cells, such as mesenchymal cells that they implant in the heart. I'm personally not a huge fan of that approach, but the idea there is where they would secrete things like growth factors that influence the heart. I don't know how effective those are. And then the third part is direct reprogramming of the cardiomyocyte. So make the cardiomyocyte within the heart more like a regenerative cell type. And so I think of the director of the Gladstone Institute, Deepak. He's looking at those type of approaches.
Yep.
Yeah, where they can reprogram the cardiomyocytes directly to become more of a regenerative cell type. So if we can, and this is just a shout out to some of the basic biology and people that study lower vertebrate animals like zebrafish. So if you can understand those pathways more and how heart regenerates in those models and what the genes are involved, we can then reprogram adult cell types to influence heart. I also, and again, like a big fan of Chuck Murray's work on using just the brute force, make the cells from pluripotent cells and transplant those. It's very interesting because those cells, even when they're transplanted, still remain immature for a certain period of time. So immature cells don't beat at the same rate as a mature cell. So the macaque monkeys that they were doing this developed arrhythmia for a couple of months. And so you have to consider that if you were to give a patient a stem cell therapy from pluripotent cells, if they're immature, and then you develop an arrhythmia for a couple of months, maybe like don't plan on exercising or stay like basically bed rest for a couple of months while those cells mature within your heart before they can reach like a full adult functional approach. But I do think that there's a lot of really cool stem cell related fields for cardiac that could be approached.
Yep, that was a great summary and just really a broad interest. And it kind of brings us this idea of reprogramming, I think, down to what will be our last question for the evening. And that is, I think, and it goes out well to both of you, whether there will be future clinical treatments for Duchenne's or other genetic degenerative diseases that are affecting muscle that might employ CRISPR, right? And use a reprogramming or a genetic replacement or modification of gene expression. And do you have a perspective on what the ethical implications of that might be? Of course, we've already gone down that path for a number of genetic diseases through CIRM, for example. Well, a number of examples. So you guys go ahead.
I got this one. So Duchenne is really one of the diseases leading the way in terms of gene therapy. So that's because Duchenne has a very known mutation. It's dystrophin. And although dystrophin has different mutations along the length of the gene that makes the dystrophin protein, it's just one. So you can design a bunch of different approaches for gene therapies and gene editing. So those are two different thoughts. So for gene therapy, they package a mini dystrophin or a micro dystrophin. So you can only package so much genetic material within something like a virus that can then infect your muscle cells. Muscle cells infect really well. And that virus starts to produce new protein that you were missing, for example, dystrophin protein. And so there's been very rigorous studies looking at what are the essential parts of dystrophin that can be made to restore these microdystrophins. And there's 3 companies doing the microdystrophin approach: Pfizer, Sarepta, and Solid. And they're all in different phases of clinical studies with that. Some of the work that I showed you today using gene editing, which is CRISPR-Cas9, uses a different approach. So instead of delivering a small mini dystrophin, they actually deliver the gene editing. So it's a guide RNA and a Cas9 that can go in there and remove the mutation, and then your body will naturally fit that back together. So my old lab, led by April Pyle, started a company called Myogene Bio, and they're pursuing a gene editing approach for Duchenne. And you could think for something like Duchenne, targeting every single skeletal muscle in the body, delivering something systemically that can go through every place is really critical. Pluripotent cells have really led the way and even being able to disease model in the first place. It really spearheaded the ability for that company to even come around in the first place. So pluripotent cells had a very important role in the disease modeling with gene editing.
All right, with that, it's just coming on to 8:30. I want to thank certainly our lecturers for this evening for some great presentations, and all of our participants for staying online. Be safe, and we hope to see you for the next lecture on June 8th. And everyone have a good evening. Thanks again.
Thank you.